very excited to have everyone here today, and we'll give all the participants a few minutes to log on. And in the meantime, I'll talk to you a little bit about how today's program format will go. I'm Michelle Shuffett. I'm the Chief Growth Officer at Radicle Science. And as we talk through the program today, you'll have the opportunity to ask our esteemed panel questions. To do that, use the Q&A button on your menu bar. And if you have any logistical questions or other types of questions, you can use the chat button for that. It looks like we have about a third of the folks logged on so far joining us here. So we will wait just one more minute to get started because we're going to start with a poll. Hello again, everyone. If you're just now joining us, welcome to Spotlight on Radicle ACES. I'm Michelle Shuffett, Chief Growth Officer at Radicle Science, and I'm very excited to be here tonight with this esteemed panel. We're going to talk a little bit with you about Radicle ACES, Advancing CBD Education and Science. But before we do that, we'd like everyone to partake in a poll so we get a better understanding of everyone that's on the line today. So I'm going to launch that right now. And there are two questions in the poll. So if you could take just 1 minute and answer these two questions, that would be great. We're about halfway there. About half of the folks participating have answered so far. So we'll give everyone just another minute or so. Okay, let's share those results so everyone can see. It looks like the majority of you all are from a cannabis company, several from a CBD company as well, some groups from research organizations, and it seems like across the audience, about half of you are somewhat familiar with Radicle ACES. That's excellent. Thank you very much for participating. And now we'll move on to our panel and let them introduce themselves.
Great. Well, thank you, Michelle. I'm excited for everything we're about to discuss today. And again, thank you to Rosemary and Gavin for taking the time to join us today. There's a lot of stuff that we'll be covering. We'll talk about some of the current regulations and some of the reasons for the lack of research on a lot of these products. Then we'll dive into the overall aggregate information that we gleaned from the study. And then Rosemary and Gavin will also have some time to discuss some of their approaches to their products as well. But a brief introduction on myself before I hand it off to our other guests. My background has been, whole career spent in healthcare, really wanting to fix healthcare systems. And so when I was in medical school, I was actually getting my medical degree, my MBA, both at the same time from UCLA. And towards the end of graduate school, I got pretty disenfranchised with all these institutions. And what became very interesting to me was the health and wellness space, because these products didn't have to go through 10 years of FDA approval. You didn't need a doctor's prescription. You didn't need insurance to cover the product because it wasn't going to cost $10,000 a month. But as I looked around, I realized there was generally a lack of data. And it's because clinical trials were really expensive. They were complex, they were tough. And so I ended up getting some seed funding from UCLA and starting the cannabis research program there. And I was there running that program for 3 years. And we grew the group into several dozen researchers, but we still encountered the same problems that kind of all clinical trials face, is that they're very slow, they're very complex. And in our case, we could not study a single one of the cannabis or CBD products that 50 plus million Americans were using. So it was very frustrating, to say the least. And so about a year and a half ago, I left UCLA to start Radicle Science. My co-founder, who some of you may be familiar with, her name is Payleen Thorogood. And her background is big data, technology, and she got into the healthcare space through an incident with a loved one that required the use of cannabis instead of opioids. And that was when she realized there wasn't data, and she wanted to create more data too. So she created the Holistic and Research Education Foundation. They became, and still are today, the largest philanthropic funders of cannabinoid research, about $6 million worth, funded at various universities around the country, but also encountered many of the similar frustrations, that these studies were slow, they were expensive, they weren't able to use the products that so many people were using. So we teamed up to create Radicle Science. And our whole mission here is to effectively democratize clinical trials so that products and brands and companies have access to trials and data. That data builds more trust, whether it's with retailers or regulators or consumers or healthcare providers. And the ways that we do that by offering studies that are, significantly faster and cheaper than anything that's come before is through a lot of technology and really just innovating around the nature of trials. And furthermore, we're also interested in eventually helping a level of precision and personalization throughout the industry as well. So eventually getting to a point where a specific individual with these demographics and behaviors desiring a specific effect can start to hone in on an exact product and exact dosage. So that big data approach exists throughout society. It doesn't really exist yet for healthcare. So that's what we're here to do. We're so blessed to be able to work with companies like Columbia Care and Trokie. And so with that, maybe I'll turn over how about ladies first? We'll turn over to Rosemary to give her introduction.
Sure, thank you. So I'm an MD PhD trained as an oncologist in Boston. And, as an oncologist, it was in the late 80s when Marinol was first approved. And it was very interesting. We were able to prescribe that for patients in oncology clinic, but patients, as it turned out, in Boston in the late 80s, early 90s would rather buy pot because it worked better. And that was my first sort of hint that there was more to cannabis than just THC, that other things were important as well, because Marinol was pure THC, right? It was synthetic THC. So I've always had an appreciation for the entourage effect and for CBD in particular. And, I've been in the the biotech business in particular for the last few decades. But I was very happy when friends of mine started Columbia Care and I was able to join them to try to study more about how exactly Jeff had said, we can help patients better. I first met Jeff at UCLA where we were trying to do some studies. And again, we were very happy to provide product, but we were shut down by regulatory concerns. So I'm very, excited to be working with Radicle to look at what we can study and come to some conclusions that are meaningful instead of, the usual branding that we all do. That's just sort of wishful thinking. And I'll now throw it to Gavin.
Thanks, Rosemary. My name is Gavin Chandler. I'm CEO of Trokie. I've spent my entire professional career in startup technology and early stage technology companies of various sorts. I've worked in environmental science. I've worked in various subsectors of big data, both on software and on hardware side. Done a lot of work in life science. And I was brought into the Trokie project back in April. And one of the things that interested me the most about Trokie was the idea of innovation of products for therapeutic applications and to really serve the needs of patients. and the health and wellness space was growing rapidly. I found that whole thing interesting. I also found it really interesting about the cannabis market in general, that the market started out as in most cases as a medical market. But when I started looking at the market, I found that the typical attributes of products that were geared towards patient care were much less prominent than the features of the market that were serving the needs of recreation. And so I got really interested in that sort of side of the space, and also really interested in the challenges that the space has in terms of just what Jeff was talking about, that the regulatory environment is extremely difficult for being able to learn about how cannabinoids can serve the needs of patients in a variety of different cases. So I was really interested in that. It seemed like a new space where there are a lot of challenges, but a lot of opportunities. And so I jumped on, and I've been really excited to work with the Radicle team. I mean, this right when I came on was when Radicle was really getting going, and the ability to do the things that I was imagining for Trokie right off the bat, in partnering with Radicle for the ACES study and for subsequent trials, is just, it's exactly what I think this industry needs in order to, as Jeff said, help to build trust, help to inform people of what's real and what's not, what's science and what's marketing, and move the industry towards more compliance, and hopefully build support at the levels of the regulatory agencies for more research, more investigation, and making the environment more conducive to those types of operations for all the constituencies that are interested in this and that it's important for.
Great. Well, thank you, Rosemary and Gavin, for joining us. And on that note, speaking of the regulations, maybe I'll briefly describe how we kind of find ourselves in this situation in general with cannabis research, but specifically what's happening with the CBD landscape. So, as most of you attendees might know, cannabis ever since the 1970s, so for over half a century, it's been labeled as a Schedule I drug. It's very difficult to study. You need special permits, special storage requirements. And for this last half century, up until literally a few months ago, there was only one registered source of compliant cannabis, federally compliant cannabis. And it was a farm at the University of Mississippi that grew cannabis for studies. So as a researcher, you'd have to get a DEA license, have a special storage facility to actually research and store the cannabis, and then you're kind of stuck with whatever was grown at the University of Mississippi, which were products that did not resemble anything that Americans are using, to say the least. And in fact, they'd even have derivative products, edibles and vapes and anything like that. It was really just flour, frozen, dried flour rolled into cannabis cigarettes that you'd have the honor of purchasing from them. You'd have to pay for it. And so all these things made research very difficult in general. Now, when it specifically came to CBD, what happened with the 2018 Farm Bill is that CBD and hemp that it was derived from no longer was considered cannabis. So it was no longer a Schedule I drug. So suddenly you see a proliferation of people growing hemp as a agricultural crop and people creating CBD products as a dietary supplement. And now the DEA was no longer involved in this process. What then emerged is a separate set of regulatory conflicts, so to speak. And so this is where the FDA stepped in saying, CBD cannot be sold as a dietary supplement. And that's because a pharma company, GW Pharmaceuticals, had already begun substantial clinical research as a CBD as a drug before it was widely sold as a supplement. And so this has been the main point of contention that has led to a lot of confusion in the sector. And so for, say, universities, when they're approached by CBD companies wanting to do research, they go, well, the FDA says this has to be studied as a pharmaceutical drug. So instead of supplement type research, we have to basically have you go back to the FDA, register as a investigational new drug, then you can come back to us and do studies. But that is a process that is really reserved for pharmaceutical companies. And so for all those, and because universities take federal funding, they're not really willing to deviate an iota from the norm. So that's how we find ourselves in this situation. 50 million Americans are using cannabis or CBD. Some are getting their cannabis, or the state legal programs in 30 plus states are providing cannabis. And then through the 2018 Farm Bill, these hemp-derived CBD products are serving tens of millions of Americans as well. And for the most part, research organizations in this country are kind of prohibited from running meaningful human studies on either of those products. So that's the conundrum we find ourselves in. And that's why it's exciting that when we came into this space, the first place that we wanted to actually conduct more studies on was CBD products, and specifically CBD products that were already in the market, and using what we call a real world evidence approach, which is not just, it's across the entire healthcare ecosystem. People are much more interested in real world studies. So this is what's happening to you when you're using the product in your own accord, in your own home. What does that look like? As opposed to an artificial clinical trial where I have you come into my hospital and I make you use the product there. And I assess you on the spot. How's your pain right now? How's your stress right now? So that doesn't really resemble how people are using these products and what outcomes they're experiencing. And so maybe this is where we'll shift gears a little bit and talk about some of the design elements of ACES. If we can pull up the slide, Michelle. Great. So when we designed the study, we wanted to first off make sure that it was there was enough time for it to be able to capture meaningful data. So we thought 4 weeks was sufficient because based upon what a lot of folks have been experiencing in the real world with these products, they were reporting anecdotally seeing effects generally within a month. So we set that as a 4-week period. We did make it an open label study. So participants did know that they were receiving a CBD product. And so what we did was we recruited close to 3,000 people, and we randomized them to either receive a CBD product or to be on the control arm. And it's called a waitlist control, where we basically say, hey, you will get a product as incentive for being in the study. You're just gonna have to wait till the study concludes, then we'll give you the product at the end. So they basically serve as a no treatment control group. And therefore what you can do is, you take a bunch of folks and you see those that receive CBD versus those that do not receive CBD, how do their outcomes change over a 4-week period? So we recruited about 3,000 participants from all across the US. A significant amount were actually people living in rural areas, which was really nice because they're very underrepresented in clinical research. And to be eligible for the study, there were pretty minimal inclusion-exclusion criteria. Again, we wanted to be as representative as well as inclusive as possible of folks, because those are the types of people buying these products, right? And as long as they endorsed having a symptom of anxiety and or sleep issue and or pain, they were eligible for the study. So we had 13 brands participating, and that's how we ran the study. Now, it was also nice in that, when we ran this and to this day, it's by far the largest randomized trial of commercially available CBD products. So we were really excited to do that. Now, the main outcome that we were looking at was actually change in well-being. And we'll talk about in a second how we assess that. But we thought that was a really holistic encompassment of how people are benefiting from these products. So that was our primary outcome. That's what the study was powered for. The secondary outcomes that we were also looking at were how their pain, sleep, or anxiety changed. Fundamentally, we believe that if these underlying symptoms change, you'll capture it in the improvement of their well-being. But we also wanted to specifically capture changes in these underlying symptoms. And again, we're comparing them to the control arm. And what the beauty of having the control arm is you can see someone who has pain or has sleep disturbance that doesn't receive CBD. Maybe some of them naturally correct. Maybe some of them don't correct. And this way we can kind of look at how, had you not received the CBD, what would have happened to your pain, your sleep disturbance, your anxiety, etc. So that's what the control group allows us to compare. So if we take a look at the next slide, we can see the ways that we measured those outcomes. So some of these indices were developed by the World Health Organization. Some of these indices were developed by the National Institutes of Health. But what's beautiful about all these outcome measures we use is these are validated instruments. So they have been used in countless other studies, and they've been shown to reliably track these underlying symptoms. And furthermore, they are sensitive to change, and we know what a meaningful change means, right? So if you change a certain amount of points on one of these scales, there's been enough literature and studies to know, is that a meaningful change in your anxiety, your sleep, et cetera? And we also tracked side effects in this study as well. And we'll be talking about that, I'm sure, later on this talk. Now, this was not explicitly a safety study. We were not drawing blood from people to check, their liver function, things like that. But it was self-reported adverse effects. These were all the brands who participated in the study. You'll notice, Columbia Care and Trokie there as well. And here is a overlay of all of the different products and their characteristics. So you can see we had form factors, we had pretty much everything under the sun in terms of form factors, and there was good diversity by dosage, by as well as the spectrum, isolate, broad, full. And so you can kind of get a sense of the diversity of products that we wanted to include in the study. Now, all the products before being allowed into the study had to demonstrate that their manufacturing practices were up to CGMP requirements, as well as they had to demonstrate batch numbers, as well as a certificate of analysis of what was actually in the products prior to being allowed to participate in the study. Like we mentioned, we had representation all over the country. One thing I will say is that, traditionally women have been underrepresented in clinical studies. So in ACES, we made especially sure to get strong female representation in our studies, and we actually overcorrected, had at this point a unbalanced ratio skewed the other way. So significantly more women than men participate in our study. Now, if you look at surveys of CBD users, they do tend to skew female. And if you look at who makes purchasing decisions in the home around health and wellness products, it usually is the women in the household. But this is something that we weren't anticipating, but we are, even subsequent studies that we've run since, we have brought that gender ratio back to even, which is what we wanted. We wanted even representation of male and female. Some of these we can probably jump into throughout the discussion. Let's see. Yes, okay. So here were some of the key findings, and I think we can then start diving into some of the discussion amongst the other panelists. But when we looked across all the 13 products versus the control arm, use of being assigned to any one of the 13 products, there was significant improvements literally across all the health outcomes: well-being, anxiety, pain, and sleep disturbance. Side effects were rare, and we have a slide talking about what they were, and generally mild. And when we looked at different characteristics of participants, whether sex or age or prior CBD use, being a completely naive user, a intermittent user, or regular user, that didn't seem to make a difference in terms of the outcome experienced after they received one of these products for 4 weeks. Interestingly, when we looked at spectrum, so when we took all the isolate products and clustered them, and we took all the broad spectrum and all the full spectrum, and we looked at the health outcomes, we didn't see any statistically significant differences. They all performed similarly when bucketed into those categories, which is interesting because there is this discussion of entourage effect and herbal synergy and whatnot. And another thing that we did find that was very interesting is that products that actually had lower doses of CBD per serving. So we instructed the participants to follow the serving suggestions on each of the products. So some products had, so the products were the manufacturers behind the products had different suggested usages. And what we actually found is when we started clustering the products by a recommended serving size, the products with lower doses of CBD per serving actually outperformed higher doses in some instances, especially around well-being, anxiety, and pain, not so much for sleep. So that was a really interesting notion because generally you hear people think, more is better. And if you look at some of the pharmaceutical clinical trial data on pure CBD for things like epilepsy, they're using 500 milligrams, 1,000 milligrams. But there's also, if you look at animal studies on CBD, they've demonstrated this interesting dose response curve where there are certain moderate doses of CBD that actually outperform higher doses. That hasn't been studied extensively in humans, but that phenomena has been seen in CBD in mice, and other drugs exhibit that phenomena as well. So with that, I'm thinking maybe we will pause there, and I'm sure we'll dive into some of these other aggregates, but that way we can pause there, and I'll let Rosemary and Gavin speak about some of their products, their experiences, their approaches, and then we can open it up to Q&A, and then we can dive into some more of the results from ACES.
Excellent. Rosemary, would you like to jump right in?
Sure. So I had mentioned before, we wanted to know more about our products in particular, and I'm sure that Gavin and others did too. So, there's CBD as sort of a class, but, most of us don't want to necessarily be lumped all together. So, and I think that the study enables us to do that. We're able to actually see your results and how you stack up compared to some of the other brands, even though they remain anonymous, right? So you can see what quartile you're in. And, I think that this became really a way for us to want to understand how much of what we're saying about our product is supported by information versus not, and also which formulations might be more interesting or more problematic than others. Okay, so that was sort of our goal here. And I'm not sure if we wanted to talk about the slides now or later, but
happy to pull them up for you now if you'd like.
Well,
yeah,
I think, yeah.
What do you think? I think that probably makes sense. Yeah, that way people aren't on pins and needles. Probably will lead to more discussion.
Well, we'll lead to more discussion because, I'm going to give you, right. So, so we used our platinum brand CBD, again, the product branding, it looks good. We think, it tests well, but what is the feedback on how it works? Okay. And like all of you, we have the next slide, we have tons of feedback. Okay. And, we have chat rooms, we have Twitter feeds, we have all sorts of things. And, these are all actual quotes from actual customers. But, it begs for more, right? I mean, everybody says, gee, it works great. Some of them say, can I get a coupon, a discount coupon for more? But this isn't enough, right? The sampling feedback that you get on your products really isn't enough. So that's one of the reasons we were very interested in working with Radicle on the ACES study. The next slide gives you sort of one of our particular questions. Again, what is the reality besides the efficacy? We also wanted to know the adverse events. We wanted to know a little bit about the taste, if we could kind of get, because we've been concerned about taste. These products do have a taste. Would it be better if they didn't? And we also have always wondered about timing of dosing. And although that doesn't come out specifically in some of these results, we're able to sort of extrapolate some of these parameters to figure out some of our results. And I think there's one more, but there's not. So what I can tell you is that looking at that, we found that the AEs, again, there were very few, as has been discussed, but they were GI, and that wasn't surprising actually, because we've heard from people that if you take a cannabinoid oil or gel cap before you go to bed, then sometimes you'll have GERD symptoms and your sleep might not be as good as you might expect it to be. And, we were very happy with our results on anxiety. We were curious why the sleep wasn't quite as high as the anxiety. And I think it's because some people will have an AE if they take some of these formulations before they go to sleep. So, that's very important when we think about how to instruct patients how to take it, because we don't just brand, we actually say, take X when. And I think that, what we learned was really very instructive. None of the comments that we had brought this out in particular, but I think that this is what we've sort of have decided after looking at some of this data and knowing our products very well. So, you helped us a lot. It's hard to hear. I can't believe I'm saying this on, one of our products might cause GERD, so people shouldn't take it before they go to bed. But the reality is we have so much education that we need to do. These effects of CBD aren't meant to just be within the next 2 hours, right? It's meant to be taken on a daily basis. So you just have an improved mood and everything should be better. So, we can avoid those AEs if people take the product correctly. And that just gets us all back to the fact that education is so important. So that was a little professorial, but, those of you who know me know I do that. And but it was very instructive. We don't want to just believe our own branding all the time. You need to see it.
Excellent. Thanks so much, Rosemary. Gavin, would you like to share a little bit about your perspective on Radicle ACES?
Yeah, so Trokie is a very young company, and so we don't have near as much of the feedback information that, say, a Columbia Care has for their products. And so for us, we were looking to glean. It was almost everything was going to be new, right? For us, we had some thoughts and some theories and propositions about how our unique route of administration might be beneficial for patients, but we really didn't have any data to support one way or the other whether or not it was going to be, a real differentiation. And so the ACEs in general was a great way for us to ascertain if we were doing, better or worse than, the other products that were in the marketplace. But there were other really interesting things that we were interested in gleaning from the data set, and and what were patients reporting about this or that would help inform research in the future, but also could help inform potentially practitioners and people seeking solutions for wellness themselves once this information was published. So I created just a few slides of some of the interesting things that we saw. And if you bring them up, Michelle, I can just talk about them a little bit. And this sort of goes to some of the things that Jeff was already talking about in terms of dosing, where they already showed that the lower dose seemed to at least work no worse, and in a lot of cases work better than for higher doses. But we were actually curious to know, okay, if we dig down a little bit more into that and we segment by symptom or the condition that patients are reporting for, we were curious to know how does that change dosing. And in our case, we were using our standard rapid relief tab formulation, which is a CBD isolate with terpenes added back in. So it's really a broad spectrum product, but we created it a little differently than say the whole hemp extracts provide their product. And what we were interested in is, okay, if you segment by people who were reporting for the symptom of anxiety, and within the trial participants, or people in the trials that reported just sleep problems, or people that reported just pain problems, how did their dose vary? And what we found that was really interesting was that while the curves overlay and the self, and we also provided, it was self-escalating dosing. So we basically told patients that they could take one all the way up to, so many doses per day, and that they should self-escalate until they feel that they've reached their health goals, so to speak. And what was really interesting was that the people in the study that reported for only pain or only anxiety had very similar curves in terms of what they reported back in as to their daily dosing. And sleep had the sort of the broadest distribution. That one was the one with the most skew. And then if you go to the next slide, when we included the group that was reporting for all three conditions, so people, and these are often related. You know, a lot of times I read some a study where people who report having anxiety and pain will almost they're a major favorite to also have sleep problems, and you can basically see that if you pick out any two of those conditions, that you're a favorite to have the third condition as a reported problem as well. But when you add in that group that reported for all three conditions, then the dosing got very broad. So we just thought that this was very interesting to start to glean some understanding about what recommended dosages might be for given sets of symptoms or given individual symptoms. So this is just a really interesting piece of information that we were able to dig into from the raw data that was reported back from patients that were in this study. Next slide. Another really interesting question that was asked in the trial of participants was whether or not they reduced their reliance on whatever other medications that they were taking when they were in the trial. And we thought this was really interesting. So we didn't see people reporting who were only reporting for the condition of anxiety or only reporting for pain. You can see that I think it was about 12% of the patients that were taking Trokie reported a reduced reliance on medications that they were previously taking for sleep when they were taking Trokie in the trial. But the really interesting one is the people that reported for all three symptoms. We had almost 30% of that particular discrete patient population. And I think in our trial or in our group, in our cohort for Trokie, that was something like about 90 patients in total reporting for sleep, anxiety, and pain. And that something like 30 of those patients suggested that they were reducing their reliance on other medications. Now, I will note that this is something that we would never recommend doing without consultation with your physician. But we just thought that this was an interesting data point that came out of the trial that can speak to maybe other research that can be done, or just at the very least what patients are reporting and what they're experiencing when they're put into this type of a trial format. So you can go ahead and go to the next slide. So another thing that we wanted to do was sort of going along this dosing question was there a relationship, knowing that the trial seemed to show that lower doses worked as well or even better than higher dose? Was that affected by other characteristics of the population, in this case, body mass index? And interestingly enough, there was almost no correlation. So that basically the weight or body mass index of the patient did not seem to affect how much they decided to take in a self-escalating pattern for our particular cohort of patients. You can go on to the next slide. And going along on this same theme, we were wondering, okay, well, does body mass index change how the effectiveness for how a person was perceiving their change in well-being? And once again, a person's body mass index did not seem to correlate with how well the product seemed to work for them in terms of their changing in their perception of their own well-being. So I think that's the last slide that I had, Michelle. So we just thought that these were all really interesting other outcomes that we could look at, and this is not meant to be statistically significant or look at any of those types of rigorous analysis. It was more just, you know, what other patterns can we see in the data that can inform us of additional research that can be done, that can speak to things like dosage, which can then speak to what would be, you know, relevant from how we advise people who are seeking to use these products for supporting their health, and then maybe down the road, what information can we provide to clinicians, or the industry can provide to clinicians, not so much Trokie, but just as a whole, what information can be provided via literature, through further research to clinicians when patients are asking them, hey, I want to use this product. What should I use? What should I use it for? How much should I use? All of these things, we're writing on a blank slate right now. There's just hardly any of this data available for within human clinical research.
Great. Well, thank you for that, Gavin. And maybe now to kind of wrap things up and start opening up Q&A, I will talk about the last bits of the ACES study. And so Mish, maybe you could pull up the slides maybe on the methods. Now we can talk about the methods. I don't want to get too deep initially. Wonderful. So we did use linear mixed models, and this accounts for different differential rates of attrition amongst different time points, amongst different product arms. It's also really good for longitudinal data as well. So being able to compare at each time point throughout the study, the active treatment group with the control arm, and also factoring in differential rates of attrition between all this. So we were comparing every product to the waitlist control. Then we started clustering products based off of characteristics. We mentioned dosage and spectrum. And when we did that, and then there was a Q&A where was this qualitative or quantitative? No, this was based on the suggested CBD dosage per product. So this is almost like an intent to treat analysis. We found that there were, for certain conditions, like well-being and anxiety, for instance, that the lower dose per serving products actually outperformed some of the higher dose ones. So clearly something else is at play here. And then, like, to reiterate what we said about the spectrum, when we clustered by broad, isolate, or full, the products when clustered that way all performed about the same. There was no statistical significant difference. And then the last thing that we also mentioned is we compared all the patient characteristics, looking at these three-way interactions. Again, we did not find that your age or your gender or how much CBD you've used in the past impacted your outcomes that we observed in the study. And speaking of those final aggregate outcomes, if we go to the next slide, this is where, so we talked about that, here we go. So what we found was that the largest improvements were observed in the first week of using the product. And those improvements still continued a little bit till the end of the study, but the delta was very different. So you can kind of think of it like this, and then tapering off after that first week. So they still improved throughout the 4-week period, but the largest improvement was observed in the first week. There was about a third breakdown between people who noticed some effect within an hour, people who noticed an effect within hours, and people who either didn't notice an acute effect or noticed it, in the window beyond that. What we saw was that there was a significant improvement in their well-being score. There was almost a 71% improvement. Again, this is the aggregate data across all the brands, that there was a 71% improvement in well-being. And when it came to anxiety, 63% experienced a clinically meaningful improvement. So again, because we use these standardized, validated outcome measures, we know what a change in that, for instance, GAD-7 anxiety score, what that means. And clinically meaningful means that it is something that is significant and palpable and is really making a difference in that person's quality of life, for example. When it came to sleep, 61% experienced a clinically meaningful improvement, so slightly less than the percentage of people who experienced it with anxiety. And then when it came to pain, it was about 47% of people who reported pain at the beginning of the study, by the end of the study, had seen a clinically meaningful improvement. So in the aggregate ACES study, we see that compared to the waitlist control and seeing these clinically meaningful improvements, people are seeing significant benefits across these various domains. And then is there...
So Jeff, that might be a good point to pause. One of our listeners asked if you could talk a little bit more about the waitlist control and explain what that means.
Sounds good. Someone also talked about, okay, so here's how it worked. So the waitlist control so everyone expresses interest in the study, saying, I would like to be in the study. I'm willing to answer questions in exchange. I'll be sent free product. And when you sign up for the study, you have a chance of being assigned to one of the products to receive it immediately, or assigned to the control arm where you receive the product at the end of the study. So you're on a wait list to receive the actual product. While you are on that wait list, we're still tracking your health outcomes. We still ask you all the same questions about who you are, what's your health status, do you have pain, sleep, anxiety, and then we're still administering those same validated outcome measures. And so while you're not getting any CBD product, you're answering these questions. And at that same parallel time, there's another group of people who receive their product. And now they're taking it and we're tracking that as well. So what this type of a control group does, it allows you to compare a few things. Number one, it allows you to see what is the natural course of some of these symptoms and conditions. Do they naturally resolve on their own? And so what you're looking for is the delta, right? If pain naturally resolved on its own in most people, then you can look in the treatment arm and see, oh, okay, was their resolution of pain very similar to how the control group just naturally resolved? Sometimes things go away on their own. They don't need a treatment. It also allows you to correct for what's happening in the macro environment. So I don't want to date this webinar, but there's some stuff happening in Europe right now. There's a potential war brewing. Russia invaded Ukraine. Maybe everyone's really anxious. And if you were studying Ukrainians, you would suddenly maybe see a spike in their anxiety. And you're like, is the product causing that or something happening in the macro environment? Well, if I look at the waitlist control and they also had a spike in, say, anxiety or sleep disturbance, clearly there's a shared macro environment going on. So those are what it allows. Those are some examples of the confounding variables that this design allows you to account for.
Excellent. I think there's a couple of other questions here along similar lines there about how did Radicle find and recruit the participants, and what were some of the exclusion criteria?
Sure. So this was a completely virtual study, 100% virtual. The products were mailed to participants. Participants filled out information either via their mobile phone or via email. We recruited people through completely digital means. There was no clinical trial sites, and we actually we partnered with some consumer research networks. And so they had a really large panel of Americans throughout the country. I basically said, we want, we kept speaking of inclusion, exclusion criteria, the main criteria was you had to be, 21 and over, have access to the internet, and have a mailing address that can receive product, and endorse one of these symptoms of pain, anxiety, or sleep disturbance. Those are the main inclusion criteria. As for the exclusion criteria, it was screening out anyone who was pregnant or attempting to get pregnant. It was screening out anyone who was definitionally a heavy alcohol consumer, anyone who had a history of liver disease, for example. I can pull up the exact list, but those are some of the main exclusion criteria that we were using. I think it was also, had you used a CBD in the last, in the very recent history, you were also excluded. But if you used it in the past, that was okay, as long as you weren't a current active regular user at the time that we were screening you. And folks didn't know how to answer the questions to get into the study, right? So it was hard to kind of game through all these screening questions.
Excellent. I think this one is a good one for Rosemary. How do you know that the CBD product is causing the effects that are being measured?
You don't. It's a very good question. Again, I've done clinical research my whole life, and you try to control for as many variables as you can, as Jeff said. All we can tell you is that that the background noise of what was going on with these patients, I think that people from the wait list compared with itself, I don't think was terribly different. I don't think there were a lot of differences within the wait list itself. When people were in the control, and I think that it's very hard to do that. You really can't. You're assuming that people understand to some extent that when they're on a study, you're trying to control variables and minimize them. And so they're not going to be introducing new diets. They're not going to be trying to go to bed at a new time. But, you know, you're absolutely right. You can't control for everything. You know, that's sort of one of the prices that we pay. But again, the downside is when you look at the end that they were actually able to get into this study and enroll, that's fabulous. You know, no one company could do that on their own. So the ability, even though there were these caveats, to have this large number of patients participate in one study with the same endpoints was fabulous.
Yeah, and to add one thing to what Rosemary said, that's why this was what we call this is a real world evidence study. So in many ways, it was trying to mimic as much as possible and capture what's happening to your average person who walks into a store and picks up a product. They're not having someone come after them saying, hey, don't do anything else to your life for the next 30 days. Don't change your diet, don't change your exercise. Like, you know, the world is messy, behavior is messy. But what we do see is in these thousands of people who were naive to these products and then use them, and we kind of systematically were just tracking what was happening to them, we see, again, meaningful improvements in these domains. And so this, again, the goal of this study was to kind of capture in a very systematic way what is likely happening to the average consumer who is ordering a product online or walking into a store and having a first kind of regular usage of a CBD product.
Excellent. Thanks, both of you. Gavin, there's a question that came in for you. In the Trokie arm with sleep anxiety and pain patients, did they discontinue their prescription dose altogether, or did they just simply reduce the daily dose of their prescription products?
Yeah, that's a great question. The answer is we don't know the answer to that question. It was a binary question in the trial surveys that basically was did you decrease your reliance on your other medications, yes or no? So all we had was the number of patients in our cohort that reported that they either did or they didn't. I'm actually very curious to see when Radicle publishes the manuscript of the aggregate data, if that's something that one of the data points that they're going to address, so that we can understand, you know, did what we see with our patient group extend to the or with our participants that were in the Trokie cohort, did that extend to all brands? So that was like one I'm really kind of curious about.
Yeah, so the balance with any of these studies is you kind of want to ask as many questions as you can without giving survey fatigue. So one thing that we did was we made sure, and this answers one of the other questions I see in the Q&A, we made sure to keep these surveys as short as possible. There's some quality of life instruments that are 20, 30 questions, you know, and so we wanted to make sure that we weren't putting people through that, because then you just have people mindlessly pushing questions. So when we were tracking dosage, it was actually a daily question. It wasn't that, you know, over the last 30 days, how much were used? It was always, hey, yesterday, how many servings did you use? And that's how we tracked dosage, for example. Side effects, same thing. We were tracking side effects at both the daily interval. You could report it daily, and at the end of the study, we also asked them to reflect on side effects experienced. But in some instances, for instance, what we just mentioned with Gavin, as much as we wanted to ask additional questions about that, we didn't want to saturate them with too many questions. So we only have this binary, did you reduce the medications you were using to treat these symptoms as well?
I thought a good question that came up, Michelle, that I saw before was somebody was asking the definition of well-being. So I thought talking just a little bit about what the WHO-5 well-being index represents, and it's essentially a series of 5 questions that range that it's how I have felt cheerful and in good spirits. I have felt calm and relaxed. I have felt active and vigorous. I woke up feeling fresh and rested, and my daily life has been filled with things that interest me. And then you rate each one of those questions are all the time, you know, most of the time, some of the time, little of the time, almost never, and then never. And then that creates a score for each one. And then you multiply by that by 4, and you get a score of 1 to 100. And that's the WHO-5 well-being index.
Excellent. Thanks, Gavin. in the time that we have left, I think I have one more question I'd love for Rosemary to tackle, and then there's a great question that would be perfect for the three of you to close the program with your thoughts on. So Rosemary, and I'm going to paraphrase a little bit here. The fact that the largest effect was seen in that first week begs the question of whether that effect remained constant over the trial or whether it slowly went down over the trial. So this person wanted to know if there's some sort of endocannabinoid system saturation explanation going on here.
I think that people just notice things when they're new. Again, I don't have a terribly scientific explanation other than that. What I can tell you is that, from the Epidiolex experience, the endocannabinoid system with regard to CBD is very mysterious. It's an agonist partial, and it's a partial agonist antagonist situation. So, I think that that book has not been written yet. That chapter has not been written. So I think that there's still a lot for us to understand about CBD binding and actually, how that works. And again, that's very relevant with regard to the broad spectrum, full spectrum, and isolate, right? What other things you have in there. So that's complicated. What I can tell you is, based on what we know about Epidiolex and how much you need to see a pharmacological effect there, I think it's very unlikely that we're saturating anything at the doses that we used in this study. I think that we're seeing a pharmacological effect, when we're using those very high doses. Here, we're somewhere between physiological and pharmacological, I think, right? So again, I don't think, but we still need to understand so much about, how CBD actually works in the endocannabinoid system. It's still not fully explained.
On that same point, one of the questions, if we still have a minute, one of the questions was that most of the effect was seen in the first week that you reported on, and they asked if there was sort of a diminution of the effect from that point forward. And if I remember from the data, it was just the opposite. Actually, there seemed to be a continual slow additional improvement over the subsequent 3 weeks. So that's sort of another interesting feature of what we can glean from how this is working in the real world, at least as it pertains to the outcomes that were being measured and the instruments used to measure them.
That's exactly it.
Excellent. Well, that's great segue to the final question in our last couple of minutes here. And I'd love for each of you to weigh in on this. And the question is, what's your conclusion on how these improvements compare to the published results that are out there? And then I'll add a little bonus there. What does it mean for the industry as a whole?
Yeah, so effectively, the beauty of using these validated survey instruments is we can now compare this to other things that have been studied. So, you can go look at other studies that have used the GAD-7 measure and see how does mindfulness affect that? How does certain medications affect that? And then you can look at the percentage of people who saw a clinically meaningful change in their GAD-7 score on blank drug or blank intervention. Then you can see that number that we just reported on the aggregate data. So that's how you can use those in comparison. And real quick, there was a question about missing data points. That's the beauty of the linear mixed model, is that as there's differential rates of attrition, it's actually able to factor all of that in. Again, I'm not a biostatistician, but we were basically using what is the gold standard in academia for tracking longitudinal human trial data that using something like a linear mixed model.
Excellent, Gavin.
So I was following Jeff. Repeat the original question again.
Just your thoughts on the overall conclusion of the study. And
and how it works with versus OTC products. Well, obviously, without direct comparative, trial data, it's tough to make any inferences, right? But what I think that we're seeing is that this data set, one of our partners said the rule is any good clinical trial deserves 3 or 4 more, right? And so I think that's what the picture that's being painted here is like, okay, now that we've got some of these, we've got some of these data sets to show efficacy from this trial. And you certainly can go and you can look at other trials that use the same validated instruments and compare the outcomes in those trials if they were for OTC meds. And you can at least get a relative comparison. But what I'm really excited about is the next step, right? The next step is using the Radicle Discovery platform to do randomized placebo-controlled blinded studies, to use the gold standard for looking at efficacy, for looking at the magnitude and seeing if there's statistical differences between the placebo and these cannabinoids as active agents. So right now I'm saying I'm saying that this data justifies going to the next step. And from what I'm hearing from Radicle, that there are several brands who are right now considering putting an active control of an OTC drug into their discovery trials. So if they're studying cannabinoids or CBD or some other minor cannabinoid for pain, that in addition to the placebo arm, that they want to put in an ibuprofen arm. And I think that that's, all of this is leading to that. We're seeing people starting to jump on board to say, hey, now we've got a real tool for doing real research that we can show to real scientists and real clinicians that are looking for data that meets the same standard that is being used for every other therapy that they are recommending to their patients. So my thought is, yes, we can go in and we can make comparisons with this data and other trials that use similar instruments for OTC, say analgesics. But I think the stage is set for the next phase, which is a whole set of more rigorous trials that are going to answer those questions maybe directly.
Excellent. Thank you, Gavin. Rosemary?
So I'm also excited about what we can do next, but I sound like the most marketing person, I think, on the panel, but the reality is that, I think this is very helpful for brands to be able to sort of perfect their product. I mean, that's what I see about this is, people, we don't have an isolate. Okay. But I'm just saying, there's differences between some of the effects and they don't all follow. I mean, the three things don't all go up and down at the same time, right? The sleep, the anxiety, and the well-being. So I think that, it helps people sort of home in on what they might want their product to look like in a perfect world and how they may want to do that before we do research. The thing I'm just always very careful with, you can spend millions of dollars, tens of millions, hundreds of millions on research, but you have to ask the question right first, and you have to have perfected the drug candidate first, right? So I think that that's sort of where I think we need to use this data and try to help make the products the best that they can be, and make the data that we do in the randomized studies meaningful. So that's what's exciting to me.
Wonderful. Thanks so much. Jeff, would you like to say a few final words and we'll close everyone out right on time here?
Yeah, so I think the final thing I'll say, and this kind of echoing what both Gavin and Rosemary said, is this is just the first shot across the bow. There is a lot of work that needs to be done. There's basically a century of research bans that we are now having to overcome. So it's amazing to work with brands and companies like Trokie and Columbia Care. Radicle's barely a year old, so we're just getting started. We're pretty excited that, last year we studied, between all our studies, we studied over about 7,000 people in our cannabinoid trials, which is more than all global cannabinoid trials combined. And that was when our company was still a startup and was only, less than a year old. So we're excited, and there's just more to come. So thanks for tuning in. And it's amazing to be at the forefront of this. It's just waking up super excited every day and just being able to work with amazing people. So thank you, Gavin and Rosemary, for joining us. Thank you, Michelle, for hosting and organizing the webinar.
Excellent. And thanks to all our participants. Hope you have a wonderful evening, and please take a moment to fill out our survey at the end of the webinar. Have a great night.
Thank you.
Take care.